Mushroom lovers, Michael here, and I am talking to Logan, who is going to share all about Mycoforestry. Now, Logan is a feral forager, herbalist, permaculturalist, and amateur mycologist, born and raised in the coniferous forest of the Willamette Valley. He studied herbalism and botany at the Columbine School of Botanical Studies, astroherbalism at the School of Evolutionary Herbalism, and has a BS in horticulture from Oregon State University. He grew up foraging wild plants and mushrooms around his home in Nordy, Oregon. He has worked extensively as an herbal medicine maker, wildcrafter, medicinal herb gardener, teacher, herbalist, and ecological landscaper. He owns his own small herbal products and consultation business, Nodi Botanica, that specializes in local sustainable wildcrafted mushrooms of the Pacific Northwest. He also works at Bastyr University as a gardener in the medicinal herb gardens and manages their Sacred Seeds Trail. Welcome so much to the summit.
Thank you very much, Michael. Happy to be here.
So you're going to talk to us about microforestry. Talk to us, give us like a little bit of an overview. What does that mean?
Yeah, so microforestry is essentially just looking at our forestry practices and the way that mushrooms really interact with them and how we can use them not only for their mycorrhizal network and how they benefit the living trees, but also with how they help degrade things, increase pathogen resistance, and like they just have such a huge myriad of benefits. So I think just really getting a deep understanding of the ecology of mushrooms helps us to have more resilient forests. So I just really see it as the symbiotic relationship. And so I'm hoping to enlighten you all a little bit on how we can use this philosophy when we work with our forestry ecosystems.
Okay, cool. Well, that's going to be very interesting.
So this is a really exciting topic because I really feel like when we're looking at our forestry ecosystems, it's important to look at it in a really holistic lens. And often we just get really focused on just what's the tree, what's good for the trees, how can we have more trees? And like we're always doing all these forestry replants, but we're not thinking about, well, the tree's not just living there by itself. It has like a myriad of other organisms living there with it, you know, from viruses to bacteria to all the animals in the area. And I think fungi is just like a really key part of that. So I think we just take a little more time to understand the role that fungi play in these forestry ecosystems, then we can really just have a lot of benefits. So I'm just going to kind of go through some of the different ways that we can use fungi. You know, one way is we look at them, they have symbiotic relationships with a lot of these trees. They can help decompose a lot of the stuff that's on the forest floor, things that are dead and dying, just like bringing it back into the soil. Then we also have the parasitic ones and like, you know, learning how to balance those relationships of ones that we see outward that look beneficial and then ones that seem not so beneficial, and how we can really create stronger, more resilient forests who can handle a little bit of parasitism here and there. So yeah, I'm just going to kind of come through this like a very holistic lens and just like, you know, making sure that we look at fungi through this. So I guess I'd like to start a little bit about telling you what my background is and why I'm talking about microforestry to begin with. So I grew up in the Willamette Valley in Oregon. My hometown is called Noti, Oregon, N-O-T-I. And it really was a logging community. It's like when I'd go out and wait for the bus stop as a kid, you know, it's like I could see there was always like logging going on up in the areas. You know, there'd be log trucks driving by when I'd be waiting for the bus, and I'd go like this, and you'd hear them like honk, honk back at me, you know. And then I was also a wildcrafter. My parents would take me out foraging wild mushrooms, and really the best place to do that was out on the logging roads. So we were riding around on all these Forest Service roads. So I really, from a young age, I got this like deep look into like what the forest ecosystems looked like where we'd find our mushrooms, like how old were the trees that we needed, what were the understory plants like, you know, and just noticing like, you know, after an area got completely cut down, like how long it took for things to grow back, what was like the forestry ecosystem like. So even at a very young age, I was like very tapped into like how forests were being managed around here, because Oregon's just like has like really big logging industry, and we also have a lot of old growth too. So it's kind of like seeing those two ecosystems going side by side. Yeah, and then as I got into high school, when I was about 16 years old, I got a job as a wildcrafter too. So I was actually doing this for my work, where it took me out to a lot more diverse areas. So I'm like, oh, you're harvesting morels, going out into the ponderosa pines and noticing how the fire ecology, you know, when the fires would come through and grow morels, how when we get these really intense fires or just really burn like really deep, it would kill off like the areas, like you'd always look for morels in the areas where it'd be like a light fire going through, just a light brush fire would just kind of brush up on them. And so, and then we see a lot of ponderosas would be the second most common tree that we'd see logged and for the logging industry, with Douglas fir being the number one out here in Oregon. As I got older, around the age of 21, I started studying botany, ecology, and herbalism at the Columbine School of Botanical Studies. So this was really cool because I was getting like an even deeper look into forest ecology and botany, because we were going out in the woods and we're like keying out all these plants and we're looking for these rare and sometimes common medicinal plants, and just like looking at the stands of them. How are they? How strong are they? How resilient are they? What's the forestry ecosystem like around them? And just getting this idea for like the diversity of these areas, and just really getting a look at lots of diverse ecosystems and really tapping into what the plants are there. And then I got, like Michael said, I got my bachelor's in science at Oregon State University in horticulture. And that kind of took me into like the more of the invisible world of fungi, I guess you could say, because, you know, I'd always been a harvester and a wild forager, and, you know, I was a big fan of stemets at the time, so I knew about, you know, the mycorrhizal networks, the symbiotic fungi. But in horticulture, I was studying plant pathology, and we were actually like taking a look at fungi that you could not see out in the area, not the ones that don't grow into mushrooms, but the ones that live inside of plants, you know, the endophytic fungi that live inside the plants that you could only see under a microscope. And so that really like opened up my world to the whole invisible world of these fungi that, you know, sometimes they just like appear, like if you look at a leaf and it's just like, oh, there's some black spots and some orange spots, but, you know, you don't think much about it. But then you realize like, oh, no, those are the overwintering Telia spores that are the black ones, and the orange ones are the summer repeating Uridinio spores. And so it really showed me that there's like this whole invisible world of fungi, kind of looking at the more parasitic fungi and the ones that live inside of the plants. So I've kind of come from this really diverse background of looking at fungi from a really holistic lens and just how they fit into these different ecosystems, and just really watching how ecology changes and what the effect of the mushrooms are within that, and just being able to see into that invisible world. I guess I should say, like, why does this matter, really? You know, it's like, okay, mycoforestry. And to me, I see it as like one of our main drivers, you know, because right now we're seeing like lots of increased intensity and weather patterns, you know, lots of talk about climate change. People are really big into carbon sequestration and like different methods we can do to help kind of combat this. And I really see forestry as being like one of the best things we can do for this. And I'm going to talk a lot about like how forest ecosystems really help to mitigate climate change through this kind of regulation of terrestrial hydrologic cycles. So I'm going to tap into that a little more, but for now I'm just going to talk about some of the current forestry issues that I really see. So one of the main things I see is like just loss of forest in general. We've lost tons and tons of forest, and a lot of the forest that we're seeing nowadays, especially in Oregon, is third growth, fourth growth, second growth trees. So by that I mean like a second growth would mean like a forest got cut down and then it got replanted, and then that'd be second growth. Third growth is that second growth forest gets cut down and then a new forest gets planted on it. And then fourth growth is, you know, that forest gets cut down and then you have fourth growth trees. So in Oregon, it's very like finding first growth outside in the lower elevations, you know, below 2,000 feet is pretty much unheard of. I think like Opal Creek's pretty much their one remaining place. So I think we have like less than 1% of first growth forest now. So it's like when we walk around Oregon, a lot of the forests we're seeing are actually second, third, fourth growth forests. You know, these forests have been cut down many times and growing back. And we have less than 20% of old growth from what we used to have. And another thing we have to realize too is like just because it's old growth does not mean it was not once cut down before. A lot of our old growth trees are actually second growth. So it used to be a forest that was, you know, first growth, and then it got cut down in second growth, and now it's been enough time that we can call it old growth forest. So yeah, it's just something to keep in mind. We're kind of losing this natural forest succession, you know, how the trees would have normally grown had it not been logged and then replanted, and then how we chose to replant those trees really affects, you know, the biodiversity looking at. With our current forestry issues, one big one I see is monoculture. When you look at a forest that's been cut down and it hasn't been replanted, you'll notice that there's a lot more diversity there. There's going to be a lot more alders and big leaf maples. I mean, depending on where you're at, I'm speaking more from being out here in the Pacific Northwest. There's a lot more tree diversity when we cut down the forest and then we just allow it to reseed naturally. And there's going to also be a lot more evolution happening because those seeds are going to have to fall on the forest floor and have their own means of survival, have that genetic backbone and that can allow them to survive whatever conditions they're landing into. So it's kind of more natural selection versus nowadays where we have nursery raised trees that are getting planted out in these forests rather than being naturally seeded. We're having these trees that are bred by humans. So they're going through these breeding programs, with like economics in mind. We want Douglas fir trees that grow really big, strong, resilient. They'll typically look at the current pest issues that are going on. So they're looking at a particular few pathogens that are big right now, which is nothing wrong with that. But, we also have to realize it's like these pathogen pressures like evolve over time. So we can create one that's like resistant to this one, but at a certain point, maybe that resistance gene will no longer work and that one pathogen we're going for is going to evolve beyond it, or a new pathogen pressure comes in. So when we start breeding things by humans, we kind of lose that kind of natural selection that's going to be happening. And then also when we have things raised in nurseries, they're typically raised in sterilized soil. So they're not getting that kind of natural resistance they'd have. Raising something in nurseries kind of be pampered. It gets all the like night nutrients they want. It gets really pampered. It gets kept safe in these little protected environments. But, like that little tree sapling, it's going to get planted out into the big, scary, wide world eventually. And it's going to need to be like resistant to that. It's not going to be in the nursery. So it's we get this kind of different idea of what it's going to be like when we actually plant it out versus when we're raising it in the nursery and just looking at it through this kind of reductionist lens. So, I mean, not that it's to say that we should never raise trees in nurseries. It's just like these are things to keep in mind when we're thinking about going forward into the future and how we're going to have resilient forests. Another thing to think about is typically the way that forestry is done here for logging. In Oregon, we have lots of clear cuts. Where like an entire stand of trees is just completely taken out. We're opposed to that would be thinning, where you just go through the stand and you just like cut out a few trees here and there and just take what you need, versus a clear cut where it's just like you just cut out everything and just harvest it all. And then typically before they do the replant, they're going to be spraying lots of herbicides as to keep away, invasive weed pressures. After the forest's been cut down for the clear cut, they're spraying lots of herbicides to keep out the so-called invasive weeds. But then you also have the issue is there's going to be non-targeted effects, other plants that they're not going after that are still going to be affected by these herbicides. And so then we're losing a lot of biodiversity because of that. And I'm sure y'all coming to the Mushroom Summit, I don't have to tell you all that. Biodiversity equals resilience. We have more species and like a greater diversity of species out there of all different kingdoms. We just get a lot more resilience because there's not one organism taking over, and I think that's a big problem with our logging practices. We're replanting the same species of tree and it becomes a monoculture. Then one pathogen comes in there and gains resistance to whatever that gene is that's like protecting it. And then, it can wipe out a whole entire stand, thousands of acres. So I just think it's really important that we try to keep that biodiversity in our systems. And so these are some big issues I'm seeing a lot in our current forestry practices. So moving ahead, what we need with our reforestation efforts is we need ones that will breed resilient forests that can survive future climatic changes, remain resilient in the face of ever-changing pathogen pressures, better utilize nutrients, efficiently biofiltrate pollutants, store carbon, and maintain healthy terrestrial hydrologic cycles. So that's like the way I'd like to surmise what I'm looking for in these microforestry practices. So yeah, I've kind of hinted on this whole concept of the terrestrial hydrologic cycle. So I'm just going to break that down for you all a little bit. Essentially, we have our terrestrial, or like our landscape, and I just see it as like the way, so we have our ocean, and the ocean's just like this like big force of insulation. It's just this massive body of water, and water has this amazing capacity to resist temperature changes. So it keeps this nice, really steady temperature and just like having this massive insulating force, lots of moisture, and it releases this moisture into the air. So it kind of creates these like kind of warm, humid, high pressure zones. And then when they come in, they pass over the landscape. They're passing over a forest. It's going to tend to create a lower pressure zone in the forest that's going to help drag in that hot air. And then coupled with that is we have our polar nuclei. So the polar nuclei are essentially like our pollen and our spores. The pollen released from our trees and our plants, and the spores released from the mushrooms. And these little nuclei are hydrophilic. So the surfaces of them actually draw water onto them, and they help water congeal around them. So you have this warm front coming in from the ocean, then this polar nuclei reaching up, and it's just like connecting with that humid air, and it's just like the water will just like congeal around these little polar nuclei, the spores and the pollen, and help create rain clouds and help them form, and then help return that rain back into the landscape. So a lot of people back in the day, a lot of indigenous cultures say the forests bring the rain, and for a long time that was thought to be folklore, but nowadays we're actually seeing like, no, the water is being released into the atmosphere and actually having these like hydrophilic surfaces actually help bring the rain and help bring the rain back onto keeping forestry ecosystems there, and the more rain that we have in is helping to prevent drought, and it's helping to just keep this terrestrial hydrologic cycle going, keeping our aquifers full of water. So yeah, that's one way I really see that fungi can help with maintaining this terrestrial hydrologic cycle. The spores, they have this sugar alcohol around their surface called mannitol, and it's super hydrophilic. And so with a lot of our fungi is when they release their spores in the atmosphere, like I said, that it'll like congeal around there. And it's still theorized, but to me, it just seems like we are finding fungi like high up in the atmosphere, and they're super hydrophilic. So to me, it does not seem like a large step to say that they are really helping maintaining this healthy hydrologic function, helping rain and water keep returning to the land. So talking about fungal ecology in forests. So as I said before, not all fungi are mushrooms. All mushrooms are fungi, but not all fungi are mushrooms. So we always associate, fungi as being like, a little toadstool, a little cap, a little stem. But like I said before, it's like there are fungi that are parasitic, some that only live underground and only associate with tree roots, some that only live inside of organisms. It's like we even have like fungi that live on us. We have fungi living in our skin. We have candida. We have athlete's foot. We have dandruff. We have good ones like Saccharomyces, which are really good for our digestive tract. And it's the same way with the forest too. It's like we have the visible fungi, that we see with the mushrooms, and then the ones that you have to see under a microscope, or maybe even some that you don't even see at all. Like when I take like a pinch of soil, like here I got a pinch of soil in my front pocket, just a minute. So in this pinch of soil, there's over 1 billion different organisms, and very few of these do we actually know what they are. And we've only recently been able to find this out due to like some of our new DNA analysis, but through like things like PCR, because normally these things would not survive in a petri plate, so we've never been able to see them. So when we're looking at our soil, we have to realize that this like super diverse system and stuff, and a lot of these fungi really are invisible. So we can look at the mushrooms, but also we have to pay attention to like some of the not so seen fungi and understand their ecology a little bit, so we can better work them in our mycoforestry and just our forestry practices in general. So yeah, as far as fungal ecology goes, there's the different kinds of fungi, different classes and what their roles are. So first we could look at is the parasitic fungi. So there was a long-held belief until the late 19th century that pretty much all fungi found growing in association with plants were parasitic. And it wasn't proved until the year 1950 that there were actually beneficial fungi out there. There was a study done with apple trees, I believe it came out of Japan, where they actually found that the ones grown with fungi in the soil actually benefited the growth of the apple trees. So you have to realize that we're kind of coming into this as newborn babies, looking at realizing that, yeah, these fungi aren't all out here to kill us. I mean, there certainly are parasitic fungi out there that we should pay attention to, but there's also a lot of beneficial fungi. So I think a lot of why mycology has been getting really popular lately is because we're just like in this whole process of deconditioning. There's like a lot of mycophobia in our culture, a lot of fear around mushrooms and seeing them as these like parasitic things and look out, they're poisonous. And I think it really comes from, this lack of recognition of that there are beneficial fungi out there. So we've always seen them as parasites until the year 1950. That was pretty much unproven that there is any benefit to having fungi in our landscape. And that leads me into our second category of fungi, which would be our saprophytic fungi, or in layman's terms, you just call them our decomposers. So these are the fungi that are going to be decomposing all the organic elements that are falling in a forestry system. We can think of like the logs and the plants and the trees that they're all falling in. And these fungi really help to break them down and build them up into soil and free up the nutrients that are locked inside. So often we'll see with our trees that there will be, when a tree first starts dying or it's in this process of dying, we'll see often like different species of fungi. Some of them, like ones I notice, are a lot of the polypores that you can see. There will be like these shelf mushrooms, one of my favorite being Fomitopsis pinicola. And Fomitopsis pinicola is a brown rot fungi. So the cool thing about the brown rot fungi is that they help with degrading cellulose. That's kind of like more of the light structure that kind of, it's like, see it more in plants too. So it's kind of that light structure, but it's leaving the lignin behind, the lignin, which is more like the structural. It's like the rebar, the stuff that really keeps it solid. So the cellulose is kind of more that lighter inner material. So it frees it up for organisms like woodpeckers to come in and start building their homes in there. And then over time, after the cellulose is all degraded, then we'll get our white rot fungi, which degrade the lignin, that really structural stuff. So then that's when that really brings the trees down and helps return it to the soil. And throughout most of history of our planet, there have been no organisms that could degrade lignin. And fungi are the only organisms out there that can degrade lignin. Yeah, and so that's why they say coal will never be a renewable resource anymore, because now we have lignin degrading fungi when before there were not. So, it still takes a few million years to make coal, but, it's like even less renewable now because we got the fungi that can eat them with their lignin peroxidases. So that's a really important role that they play, because I think if we didn't have fungi degrading lignin, we just have all these like it makes up 25% of the tree's mass. So 25% of all the trees ever not degrading, that's a lot of stuff. So it really builds up the soil and creates this like really healthy soil structure. So we really need them around. All right. And then next it comes into our mycorrhizal fungi. Yes, these are some of the favorite ones to talk about because these are symbiotic mushrooms. These are the ones that grow in association with different plants and different trees, and they really have this like nature of sharing. One thing we found is that a tree will share up to 15% of its photosynthates, which means like the sugars it gets from photosynthesizing, all those glucose carbohydrate sugars. It'll share 15% of the ones it produces with its fungal host. But in return, it gets an exchange of nutrients such as nitrogen, phosphorus, and lots of trace minerals. Especially really good with phosphorus. Say a tree gets about 85% of its nitrogen from its fungal symbiont. So it's really beneficial to have this there. And so these fungi will also increase the root surface area. So, it's like the roots, that's the tree's way of gathering nutrients, gathering water. So the more surface area it has, the more access it has to what's in the soil, more access it has to nutrients, more access to water. So when it's associated with mycorrhizal fungi, that surface area is increased like 100-fold, up to 100-fold. So that's a lot more access there. So this can improve water access 50 to 100 times more than it normally would. It can also help increase pathogen resistance. They have multiple levels for doing. I'm going to get a little more into the pathogen resistance things. They can also, the mycorrhizal fungi can also help to free up nutrients, like I was saying, kind of some of the saprophytes. They can help break down nutrients that are otherwise inaccessible to the roots, that are maybe locked up in inorganic forms. The fungi can break it down and feed it to its host. And so an interesting thing about this is, so this is all great, but it's like the tree or the plant is the decider in this relationship. So the tree, if it's looking for a mycorrhizal host, it'll actually send out like hormone signals out into the soil. One of them being strigolactones. There are these hormone signals that they'll send out, and they'll be like, I want to form into a mycorrhizal relationship with you. So only if the plant decides to make the signal is the fungi going to be over there able to do it. So there'll be like when the strigolactones are released or whatever hormone signal it is, there may be dormant spores in the soil, which is often how it happens. And when it senses that hormone signal, it'll actually start to germinate and release like a hyphal thread. It's kind of like the root of the mushroom. So the hyphal thread will go out and it'll follow the hormone signal, come in contact with the tree's roots, and then the tree will decide whether it wants to let it in or not. Yeah, so there's no trickery involved. So the reason why a tree might not decide to have a fungal host, because you heard all those benefits I gave, but plant might not decide if it already has enough nitrogen, if it has enough phosphorus, if it's not in a drought situation, then it's like, I'm not giving up 10% of my sugar for you, I want to keep my own sugar. So that's where we see it's important to not necessarily overfeed our trees, not like spoil them too much, because sometimes if they get a little bit stressed, they're more likely to form into this mycorrhizal relationship, and then they're going to have better access to these nutrients, and they're able to go deeper and deeper. So this is like looking at more long-term forester survival. So yeah, maybe not spoiling the tree so much when it's young, so it's going to want to reach out and create this. So yeah, another important thing with these symbiotic fungi is oftentimes they're obligate symbionts, meaning without a host, they're not going to be able to survive. So they really rely on these trees for their carbon sugars. So it's important that we don't, that's the big issue with clear cuts right there, is because if we remove all the trees and they're gone for a while, then all these fungi, we're going to lose that whole mycorrhizal network that was there, that was like thriving in that whole forestry ecosystem. It's going to take a lot more time for it to reestablish. And, there may still be some spores underneath in the soil that were previously associated with it, but they kind of lose viability over time. So it's really important that if we're going to be doing these different forestry practices, that we don't have long fallow periods, because if they go too long fallow, then we lose a lot of these mycorrhizal networks that would normally, if you just plant a tree and there's a healthy mycorrhizal network, then it's just going to hook right up with it and just have all those benefits I mentioned before with water and nutrition. And another interesting way of looking at it too is, as I was speaking before about the herbicides that we use to keep out the invasive plants. So if we already have that healthy mycorrhizal network there, then it's like the tree has that benefit of having that extra access to all of the nutrition that maybe a weed would have a lot more hard time competing with it, because it wouldn't be able to establish the mycorrhizal network. After we cut down all the trees and we destroy that mycorrhizal network, then it's a lot easier for some weeds to come in and just kind of take over things. Because there's a really interesting study by Kabir P, and he found that when they used soil in a nursery that had some of the same that was growing near the tree or plant that they were looking to work with, it would have this competitive advantage because the indigenous mycorrhizal culture was already there. And then also with a lot of our weeds, things like in the brassica family or the amaranth family, they don't actually associate with mushrooms. So 95% of vascular plants do associate with mushrooms, but there are few, which happen to be some of our weedy species, that do not. So if the soil's kind of hard to live in, and most of the nutrition is only accessible by having a mycorrhizal network, you get this real competitive advantage of having a strong community there, that the trees, you just plant them right into it. And so this whole idea of the mycelial network is that these symbiotic fungi aren't just associating with one tree, they're associating with a whole stand of trees. And there's association with the older trees, giving nutrients to the younger trees, and there's whole messengers. I'm going to get more into that detail into that, but I just want to mention that there's this kind of network communication going on. That's another reason it's really important to keep these healthy fungal communities going. As far as our mycorrhizal mushrooms are concerned, we have a couple different groups that I like to get into. The first of those being the Ectomycorrhizae. So ecto means outer. So these mushrooms, these fungi will associate with the tree roots and they'll create this like protective sheath on the outside of the tree. So it's creating this outer connected sheath over the roots, and it won't act, it'll go in nutrient exchange with the plant, but it does it through apoplast of the cortical cell of the root. So it's right outside of the cell of the root. It's not going actually into the cell. And it'll produce this little structure called a Hartig net that helps aid in that nutrient exchange between it and its host. Many of these mushrooms are the ones that do make mushrooms. So these fungi do make a lot of mushrooms. And so they actually have the competitive advantage of being able to spread their spores out through the air. So a lot of these Ectomycorrhizae, you'll see a lot of our edible species, like a lot of us know chanterelles or hedgehog mushrooms, a lot of our boletes, a lot of these really tasty edible mushrooms are Ectomycorrhizae. And so they're actually associating with the trees. So often when we're out foraging, we'll notice that certain mushrooms associate with certain trees because they have this mycorrhizal network. And up here in the northern climates, it's typically the Ectomycorrhizae. So yeah, so this is in comparison to the Endomycorrhizae. So endo meaning into. So rather than the ecto being outside, just having that protective sheath and kind of outside the cortical root cell, these Endomycorrhizae go directly into the root cell. They go right into the cortical root cell. And so the ones we often talk about most with our plant systems, whether it's forestry or farming systems, especially because these are the ones that are typically associating with annual plants, are the Arbuscular Mycorrhizae. So these are a form of the Endomycorrhizae, and they call them Arbuscular Mycorrhizae because it comes from that the word arbor meaning tree. Because when we look at the way when they make that relationship and we look inside the cortical root cell, when it penetrates in there, it'll create this little thing called an arbuscule, and it looks like a tree branch. It looks kind of like it's just this tree branch going inside there, and that has direct nutrient exchange with the inside of the cell of the root. So when they're exchanging photosynthates and nutrients going into the tree, it's coming through this little arbuscule. So the way we can see how climate will change with the Arbuscular Mycorrhizae versus the Endomycorrhizae is that we'll notice that our Ectomycorrhizae tend to be with the trees, will tend to be in the more northern climates, will tend to be in areas that have like less precipitation and colder weather. So there's like lower rates of decomposition. Those are where you're going to find your Ectomycorrhizae, the ones that grow on the outside of the roots. Where versus the Arbuscular Mycorrhizae, they're going to associate more with annual plants, as well as being in areas that are more tropical. They have warmer weather and higher precipitation areas where there's like high levels of decomposition coming on. It's good to keep this in mind when we're choosing what kind of mycorrhizal association we might expect. Although it's like trees will often they'll start out their relationship with maybe some Arbuscular Mycorrhizae, and then they'll switch on to EM later on in life, or sometimes they'll be associating with both at the same time. So it's good to keep that in mind when we're doing this, when we're choosing mushrooms that associate with our forests. Another thing with our Arbuscular Mycorrhizae, so unlike the Ectomycorrhizae, they don't produce mushrooms. So these are not mushroom producing fungi. They produce these mycelial networks under the soil though, and they create these things called ascospores. And ascospores are these big, heavy honking spores. So they don't have that benefit of being able to release spores out into the air that are going to go up into the atmosphere. Their spores are going to be right underneath the ground. They're big, they're heavy, and that is why it's really important to have hosts around for these Arbuscular Mycorrhizae, because once the hosts are taken away, that spore is just going to lay dormant for as long as it can before it just no longer viable. So especially when we're working with Arbuscular Mycorrhizae, it's really important to not have long fallow periods as to keep the colonies of AM fungi, Arbuscular Mycorrhizae going. All right, so I'd like to talk a little more about the fungal forest ecology and how these mycorrhizal fungi really fit in there. So, like I said before, these beneficial symbiotic fungi are not just associating with one tree, but they're actually connecting a lot of the trees in the whole ecosystem. So they're almost like this underground kind of communication, nutrient exchange network. So we'll find that even like a lot of our older trees will be like giving water, sugar, nutrients to the younger trees. So it's kind of like this older generation, like sitting there and allowing those who are like beneath the forest canopy and don't have that access to light, a little bit of health and nutrition. So like when the time comes and that older tree dies, its progeny will have, the necessary nutrients and sugars it needs to survive and work its way up to the canopy. So it's really this kind of older generations think the younger generations and the long-term survival. It's not this whole survival of the fittest, I'm trying to shade you out, but they're actually giving their nutrients to these younger trees, and they do it through this mycorrhizal network. So it's really creating this whole communication pathway between the mushroom, the tree, and even this mycorrhizal network will extend beyond just trees of the same species, but it'll actually go into trees of other species and other plants of different species. So it's really this like whole interconnected forest communication network. Yeah, and so we even found with a lot of the understory Douglas fir saplings, they receive 21% of their water from the older trees. That was shared through that mycorrhizal network. So yeah, it's just really showing that they really are caring. So like I said before, it's like when we have these clear cuts, we really deprive these young trees of that shared association. So, we do a clear cut and then we plant in all these young saplings. Without those older trees around, it's like those young trees are going to have to start from scratch, basically. They're not going to have that older generation looking out for them and exchanging nutrients in this whole network. They're communicating with them. Yeah, and it's also interesting too, because this mycorrhizal network also helps to warn of pathogens. So the older trees can, like if one of the trees at the edge of the forest is sensing like, hey, I'm actually getting like some fungus or some other pathogen that's affecting me, it can send a chemical messenger out and let the surrounding trees know, so they can produce like secondary, or I like to say specialized metabolites that help kick up that host resistance. Yeah, so I think this one article written by Samard really lays out what the benefits are here with this association with the older trees and the mycorrhizae with the younger trees. So it increases enhanced understory seedling survival, growth, nutrition, mycorrhization, increases their defense chemistry and the kin selection, like picking out ones that can survive the best through changing times. And then just demonstrates a collective memory-based interactions among trees and fungi. I just think that's a really cool way to just really lay out what this whole interaction means. Yeah, and just to give an example, if any of you all have ever gone hunting for truffles, then you might have heard of the rhizopogon. So for those of you who haven't heard of rhizopogon, we call it the false truffle. It's that one that when you're looking in the forest and you're trying to dig up and you think you found a truffle and you look in and you're like, it's a rhizopogon. It's not the one you want to be eating. It's not that valuable truffle. But they're really cool though, because these are actually symbiotic mycorrhizal fungi. And so they did this really cool study where they're looking to see like, okay, how many like trees are these rhizopogon associating with? How big is their mycorrhizal community with the Douglas fir trees that they're growing with? And so they found 8 individual mushrooms that were connecting 47 different trees. And then they found one single mushroom that was connecting 19 different trees. And so mind you, this is just like looking at a small slice here. There's not a lot of research out there, but I just think that's really fascinating that we can find 47 different trees associating with just like 8 mushrooms. And so that's showing like 47 trees are having this like intercommunication just with this one species. It's really fascinating. All right, so next we can look at pathogen production, ways that these mycorrhizal help with protection against pathogens. So I'd say like one of the first ones you could look at is like with the Ectomycorrhizae, remind ecto meaning outer. So the Ectomycorrhizae actually grow this protective sheath around the roots. Sometimes you can dig up plants or trees and you'll actually notice like mycorrhizal fungi, you notice these hyphal threads like covering the roots, and witness that. And they actually create like antimicrobial compounds within this mycelium that's growing around the tree roots. So you're getting this protective sheath that's providing that physical barrier. But then it also has all these different antimicrobial specialized metabolites that are going to help protect those tree roots. There's also stuff that's shown that these fungi will also even release enzymes that'll help degrade pathogens. I remember when I was doing some research for this one farmer while I was at Oregon State University for the root knot nematode, and actually found that the trichoderma species of Arbuscular Mycorrhizae would actually release nematoxic compounds and these enzymes that would degrade the juvenile nematodes' exoskeletons as well as the nematode eggs. So they were growing around these plant roots and actually releasing these compounds to help keep the root knot nematode, which would be normally boring its way into the plant roots. In these cases, I believe they were tomatoes. Yeah. And keeping them out of the system. So I think that's just really cool. And then maybe some of you have seen the nematode snare traps. So this is really cool. They're like these little like it sounds, it's like a little snare. You've seen there'll be Arbuscular Mycorrhizae and the nematode comes swimming along. Then like the mycorrhizal fungi will sniff it and like snap it, and catch it in this little trap, and then degrade it and eat it. Yeah, I should have mentioned before, but I have some notes included with this that's going to have pictures of this. So I'll have a picture of the nematode snare trap and pictures of the heart-tube net, some of these other structures. So yeah, I really encourage you to check out the notes that are going to be linked at the bottom of this. I'm sorry for not mentioning it before, but yeah, I just thought it'd be really exciting for you to see this. They have a really cool microscopic picture showing the nematode snare trap. So just wanted to mention that so you guys know what I'm talking about. It's one thing for me to talk about, but actually see like little nematode with its head cut in a little hyphal thread. It's pretty magical. Just saying, check it out. Yeah, and then another way that they help with pathogen production is they actually, when they're associated with the host, they find that the host tree or plant will actually produce more specialized metabolites that help with its own host defense, as well as the fungi often even producing secondary metabolites, specialized metabolites that help with the plant or the tree's innate immunity, their ability to fight off pathogens and having that initial resistance. So when I'm looking at doing microforestry, it's like we're worried about pathogens. It's like rather than thinking like, oh, we just need to eliminate all pathogens, we need to spray, we need to find ways to eliminate things. We can just like look at increasing the own plant or the tree's own host defense, their own innate immunity, making sure they have enough specialized metabolites and they're healthy and they have access to nutrition and fungi that are going to help them create these metabolites so they can remain strong in the force of ever-changing pathogen pressures, because we really can't predict the future and what the next pressure is going to be. So I think it's really important to have these associations. I know there's a lot of talk about CO2, carbon being released into our atmosphere, and yeah, I see this as fungi as being a really important way that we can help our forests keep storing carbon. And that's another reason why we need to keep our forests, because trees really help to sequester carbon, but fungi help aid in that sequestration effort. So one way they do this is with this amazing compound called glomalin. So glomalin is this carbon-rich glycoprotein, and it's improved the structure of the soil. So it's released by fungi, especially ones in the Glomeromycota, which is an Arbuscular Mycorrhizae. So it improves the soil structure. It kind of gives soil this sticky texture. It helps the little soil particles kind of stick and clump together. So that's like a sign of good, healthy soil. And it's really carbon rich. So it's getting more carbon into the soil, creates more habitat for microorganisms, and improves the water holding capacity. So it helps water be retained in the soil. So it's this really interesting compound to keep in there. And they find it can last in the soil for about 4 decades. So that's 40 years of long-term carbon storage. So they had this really interesting study that came out of Sweden. So they were looking in the boreal forest in Sweden. And so they found that 50 to 70% of the carbon in the forest was actually stored deep in the soil, associating with the roots and the root-associated microorganisms. So this one really took me by surprise, because you think okay, I'd expect most of the carbon to be up in the woody material, up in the plant material, or decaying on the surface, all the logs decaying there. That seems like where the most carbon, but I was really surprised to learn that it was actually being taken deep into the soil. And so I see a lot of this being associated with the fungi's ability to degrade lignin and bring all these carbon-rich materials and degrade them back into the soil, as well as glomalin. When these glomalin-producing fungi are present, it accounts for about 27 to 30% of the soil and the soil carbon that's there. So yeah, it's just a really great long-term soil carbon storage that creates healthier soil, better water holding capacity, all these things. So yeah, that's another reason it's important to keep fungi associated with their soils for that really long-term carbon storage. Okay, so I think I've given you all a pretty good idea of these different ecological roles of fungi, but now I'd like to talk more about Microforestry practices. Okay, so we got an idea of the ecology. How can we actually take this information and do something practical with it? How can we take this information and actually make a resilient forest with it and make sure our forests are associating with more fungi? So first thing is, I'd say, like, this is number one, is working with the indigenous mycorrhizae that are there. So I think just like that first thing I mentioned about planting our new trees near older trees, like I said before, because they already have that associated mycorrhizal network. They've done studies where they found when there was like giant clear cuts, that trees that were planted like towards the edge of the clear cut, more towards where like the older trees were, did a lot better than trees planted in the center of the clear cut, which I guess isn't too big of a surprise, but it's still like important to understand a lot of that reason why there's so much increased seedling growth. Yeah, they said it was like about 41% increase in seedling sapling growth when they're associated with older trees versus, grown without that mycorrhizal network. Yeah, that's the important thing, is thinking like planting around that edge habitat and doing more selective cutting, doing more thinning through our forest. So rather than doing a clear cut, if we just do selective thinning when we come back in to replant, or if we just allow the trees to naturally seed and grow themselves, then we're going to have a lot more resilience there because we're going to have that established mycorrhizal network. So let's say you don't have that option to do a thinning and you're just wanting to get some trees plants from a nursery. Like I said before, a big issue with nurseries is they're using sterilized soil. And the problem with the sterilized soil is, the forests aren't sterile. Once that tree's planted out there, it's going to be like hit with a bombardment of good things, bad things, things that aren't so good or bad, but it's just like, we need to make sure that they're ready for whatever's about to come their way. So one way, rather than using sterilized soil, is one thing we can do is we can go to these stands of trees and plants of the species that we're working with. So let's say it's like Douglas fir, and I want to grow a bunch of Douglas fir seedlings in my nursery. Rather than using sterilized soil, I can just go out to a stand of nice old Douglas fir and just grab some handfuls of soil and mix it in with my soil mix. Because that soil is going to already have that indigenous mycorrhizae culture there that's going to be beneficial for the trees I'm trying to grow or the plants I'm trying to grow. So this is like one of the most simple, basic methods that you can do. It's just making sure you have a little bit of soil from the area that has the species you're looking for there with an established colony of mycorrhizae and just adding it to those young tree seedlings. So I've done this method before with growing oaks, and I've had really, really good success with that. So yeah, I just grabbed like some soil from a stand of oaks, incorporated with my acorns. I was planting in an area where there weren't any oaks. So I know there would be a mycorrhizal network, and then it really helped like increasing that growth really well. And I've done it without that, and definitely seen how they were a little bit struggling when they didn't have that indigenous soil there. Okay, so let's say you don't have access to a stand of trees that has these indigenous mycorrhizae. Another thing we could do is look for mycorrhizal inoculants. So these are products often, so they can be in powder or liquid form, or sometimes like even when we buy soil or compost, we'll see it'll have a list of the mycorrhizae that are in there. Trees will often associate with arbuscular mycorrhizae, ectomycorrhizae, or both. And they switch up this relationship during different stages of their life, sometimes starting with arbuscular and then moving on to ecto later on. So it's important to keep this in mind when we're trying to choose a mycorrhizal product that works for us. So I don't work with any of these companies who serve mycorrhizal products or anything. So I'm not going to be like giving shout outs to any of them. But I think if we're going to look for the right species, different places where we can start. So let's say we have some tree seedlings that we want to, or we're planning on starting, and we want to inoculate them like with a mycorrhizal culture, but we're not sure if like the mycorrhizal culture in this brand that we're looking at is going to have the right ones. So one way is if we're going to look for a specific species is we can go on Google Scholar and we can just look up, like, let's say it's like Douglas fir. There's going to be a lot of research out there for a tree like that on what type of fungi associate with them. So we could say like, look like Douglas fir mycorrhizae, look on Google Scholar, and you might see a big list of species that associate with Douglas fir. So when we're looking to buy a mycorrhizal product, we can be like, okay, like it says like these species associate with it, and I'm looking at this product and it has these species, like therefore I think it would be a good one to use with that. And if you're having trouble looking on Google Scholar, it is simpler. A lot of this talk was based on after I read the book Teaming with Fungi by Lowenfeld. So Teaming with Fungi has a really quick guide, so you can see some of the few that we know about that associate with different common plants and trees. So that's another good resource. So if you're not finding in Teaming with Fungi, I say look on Google Scholar. And then, going back to Ectomycorrhizae versus Arbuscular Mycorrhizae and where they like to grow. So if you're out here in the Pacific Northwest like me, or in a more northern temperate climate with moderate rain and cooler temperatures, you're more likely to expect Ectomycorrhizae to associate with your plants. Versus if you're a warm tropical climate, with like high rainfall and higher temperatures, with like more massive, like higher rates of decomposition, you're more likely to find Arbuscular Mycorrhizae. So keeping that in mind, so if you're just looking and I say like, okay, maybe I'll just I can't find exact species, but I think like since I'm growing in a more northern temperate climate with these trees, I'm guessing probably Ectomycorrhizae might be good. Or versus if you're growing in the tropics or somewhere really hot and warm with high temperatures, then you'd be like, okay, I think like these trees are more likely to be with it. Arbuscular Mycorrhizae. And so that's when you can use a more generalist species. So generalist versus specialist. So a specialist species would be one like this species only associates with this one other species of tree, versus like the generalist species will associate with many different species. So if we're using a generalist species of Ectomycorrhizae or Arbuscular Mycorrhizae, then we're going to be more likely to have success when we're inoculating. Yeah, and so as far as inoculating goes, you can apply these mycorrhizal inoculants at all stages of growth, really. But it's best to do it the sooner the better. The sooner you get this association going, the better. So you can even just apply it to your seeds. When you're seeding out a tray, just sprinkle a little of this mycorrhizal inoculant on the seeds. If you're having the young plants or you're transplanting, I try to apply it directly to the roots. The closer these spores make contact with the roots, the more likely you're going to have germination and success and create this specialized relationship. Then also too, going back to remembering that the plant's to the cider. So if you're like putting lots of nitrogen and you're overwatering it, the plant's not likely to even like make that association because it's like, I'm getting all I need from the soil. Why would I need to make this relationship? But after a while, it's going to get planted out in the wild world where it might not have access to those things. So not spoiling our plants too much when they're young, make them work for it a little bit, make it so like, hey, you need to make some friends if you're going to survive out in the wide world. So, don't spoil your children too much. Yeah, and then there's also ways you can make your own mycorrhizal inoculants. I have not tried this method, so I'm not going to go into too much detail, but I just want to mention it. It's called the wet sieve method. So basically, you'd be going to those areas where you'd be finding those indigenous mycorrhizae, it's growing around those stands of trees. So let's say you have a stand of Douglas fir and you're trying to get some good mycorrhizal spores for that. You would just go up to the mycorrhizal stand, you'd sieve the dirt, and then you do this filtration study, kind of float out some of the particulate matter and put it through a screen. Like I said, I've never tried this before, but if you're interested in the wet sieve method and getting higher concentrations of these spores, there's a whole guide in the book Teaming with Fungi by Lowenfeld. And there's also a free online PDF. It's by Paccioni. So his name is spelled Pacioni. And like I said, I'm going to be including some notes with this whole thing and my whole 25-page manifesto on mycoforestry. So yeah, if you want to see all my sources, where I'm getting all my information from, it's all located in my notes, as well as in this 25-page microforestry manifesto that I wrote. I mean by a research paper. This thing, manifesto, sounds cool. So yeah, if you're interested in these resources, please check out those papers and my notes. Next, as far as a good method of application to increase mycorrhizal fungi in the soil is with activated biochar. So I'm sure a lot of you have heard of biochar, but for those of you who don't know what biochar is, it's essentially like charcoal. But unlike charcoal, this charcoal is specifically made in a low to no oxygen environment. So they have specialized vessels that do this. Oftentimes we'll see like we have a big fire and like all the stuff's burning on top, like a big bonfire. Some of the stuff at the bottom, it's got so much stuff on top of it, that stuff that burns down, there's in this kind of low to no oxygen environment. So a lot of stuff at the bottom will be more biochar-like. And so we can use different materials. There's a wide range of plant materials, tree and woody materials, as well as manures that we can process into biochar. And so when I say activated biochar, what I'm talking about is making sure it has a colony of beneficial microorganisms already associated with the biochar before you add it to the soil. So let's say you just buy some biochar, it doesn't have anything inoculated with it as far as beneficial microorganisms. You just throw it in the soil. It's actually going to help to like kind of suck out a lot of nutrition and make it harder for the plant to access things. It's just very like a lot of carbon going in the soil and just kind of sucks things into it. But when we activate it, so ways we can activate it is by adding things like effective microorganisms, bokashi, or just mixing it with our compost. And I usually do this about 6 weeks before I apply it to the soil. So once we inoculate it with these beneficial microorganisms, it creates like, they'll start living inside the biochar and they're creating these colonies and they create this, and rather than it just like the biochar just like sucking in all the water and nutrition in the soil, it has like these beneficial microorganisms that are going to be associating more with the roots, and there's going to be more cation exchange capacity, more exchange of nutrients going on. So it's really important that we activate our biochar, either just by throwing it into our compost or adding these bokashi or other inoculants about 6 weeks before we add it to the soil. Yeah, so benefits of biochar. So one thing it does, it increases the soil porosity. So that means like there's a greater distribution of these little pores. So like the surface, the biochar has like tons of teeny tiny pores of all different varying sizes. So pretty much a good soil structure has like huge diversity of pore size distribution, because different microorganisms like to live in different sized little pores. They like to find their little holes, their little cubbies, make their own little habitats there. So biochar just kind of creates this extra added habitat. It also helps with soil moisture retention, helps with that water holding capacity, making sure that there's more water in the soil. It helps with the aggregate stability while reducing soil bulk density. So rather than soil being like really compact, it helps like create these like little big chunks that kind of help break up the soil, break up that bulk density, and then also help with that, like I said, like good soil structure is kind of sticky, like it kind of clumps up and you get that kind of aggregates. So aggregate stability, so it's kind of creating that nice healthy structure due to the added microorganisms. It increases the soil's carbon storage capacities, because really what biochar is it's just carbon, just burnt carbon. And this carbon source can actually, I was really surprised. I'm like, I wonder how long biochar lasts in the soil. And they say it lasts for thousands of years in the soil, which really blew my mind. I've read it a few times and like it seems pretty legit that it's like this soil carbon source that just stays in there for thousands of years. So not only like when we make biochar, it's like stopping this CO2 from being released in the atmosphere, keeping our soil, and then keeping this healthier soil structure going on. So a really good term, long-term carbon storage for thousands of years. It's really amazing. And then it has this tendency to increase the cation exchange capacity. So cations being those positively charged particles that are attached to soil colloids. The outside surface of soil of clay tends to be negative, while a lot of our nutrients tend to be positively charged. So there's like this exchange that happens between roots and the soil colloids. And biochar kind of helps with this exchange capacity with the roots and the nutrients that are locked up in the soil. So that's one way that biochar can help to increase nutrient absorption. It also increases concentrations of available phosphorus and potassium. So really key nutrients, essential key nutrients for plant growth. So yeah, like I said, it also is creating beneficial habitat for fungi as well. I know trichoderma are ones that really love to live in biochar, and like I said, with the trichoderma, they're those ones that helps with the nematodes, and they're in Arbuscular Mycorrhizae. But there's like a lot of different species of fungi that can live in the biochar. So really good for creating that healthy mycorrhizal network there. Yeah, and so they actually showed that there was 41% increase in biomass in these forest ecosystems, especially with the young tree seedlings, when there's biochar present. So yeah, a 41% increase in biomass production, basically growth for younger tree seedlings and the trees in general when we had biochar in our forestry ecosystems. All right, so that really covers all the practical applications. Yeah, I just like to conclude by just thanking you for sitting through this. I know there was a lot of information there. I hope you all found it really interesting, and I'm going to provide resources at the end, so please check out the link below. I have all my class notes and everything. So if I could just like give like a debriefing, just say what I like to say is like, I really hope you walked away from this talk not just thinking of microforestry as like, oh, what products do I buy to increase like, you know, these things. Like, I really want to just like view it as more of like just understanding the ecology. Because when we understand the ecology, it's like we realize these mycorrhizal networks, they're already there, they're already prevalent. And if we already have a forestry ecosystem, the main thing is just like not disrupting it really, and just finding ways that we can just like work with it, not against it. So the first way by doing that is doing thinning rather than clear cutting. I know people have been saying that for many years, but yeah, I'm just going to keep maintaining that. And I think this extra added understanding of how these older trees like maintain these mycorrhizal networks. And if we lose them, if we clear cut them all, then it's like the younger trees are going to have that much more trouble to survive. We're going to be increasingly reliant more on herbicides and pesticides to like keep them safe. So I think it's just really just understanding that basic of ecology there is like keeping these mycorrhizal networks healthy and going. And when we do our replants, making sure that our trees are like close by some of these older trees so they can hook up with that mycorrhizal network. And then also like, you know, just like the fact that, you know, we're really like having a lot of these nursery raised trees, but like we need to be like allowing these trees to evolve naturally. There's going to be a lot more new pathogen pressures. We really can't predict the future. And even our best breeding programs are never going to be able to really prepare for the unknown. And so I think a lot of the time when it's like, it might be an unpopular thing to say, but sometimes I think we should not replant forests and we should let them replant themselves. And I know it's like, you know, it's in our best interest, like we should be growing more trees and doing, I'm not against not planting. I'm all for planting trees. However, I noticed the way a forestry ecosystem will naturally replant itself when I've seen like second, third growth forests where there wasn't a replant afterwards and that it was just like the trees were just able to go naturally. There was a greater diversity. There was so much more diversity of tree species. And then in return, there's a greater diversity of plant species. And you also realize you're creating that natural selection going on and stuff because like the trees are dropping lots of seeds and the ones that are most resilient are going to be the ones that survive long term. So as much as we can try to do these breeding programs and try to prepare for the future, I really think natural selection, letting these forestry ecosystems replant themselves is going to be the way to go. And then it's like that also comes in, you know, fungi become a big part of that natural selection process because a lot of these trees, the ones that are going to survive, are going to be the ones that make that as fungal associations. And then the older progeny of trees are going to decide which of the younger trees they want to survive. So it's really this like deep communication, and it's like they're actually deciding how they want to evolve. So the less we can interfere with that process, the better. I'm all for planting trees, but I just like to keep that in mind, how there can be a lot of benefit from allowing these forests to evolve naturally and reseed naturally. And so, yeah, I just want to keep ecology, that indigenous mycorrhizae, foremost in your mind. However, I am totally open if you're in a nursery, incorporating those indigenous microorganisms into your soil, just through adding that natural soil there. Or even like I'm not against using mycorrhizal inoculant products. I use them quite a bit, and I've had just amazing results. I think there are a lot of good products out there, and a lot of these I've seen so much benefit from when I add like a mycorrhizal inoculant to the roots of my plants or to my seeds versus when I didn't do it. And same thing goes with biochar. One of my professors, he's been working with biochar and mycorrhizal inoculants for about 30 years, and he's watched these trees grow. He said, like, this tree I planted with, this one I did without, and there's lots of research out there showing pictures of how much benefit it gets, you know. So yeah, biochar, mycorrhizal inoculants, really good. All right, and then just my overall message from this is, you know, reforestation is our best tool for combating all the climate chaos we're seeing going on out there. Not only for maintaining that terrestrial hydrologic cycle and storing carbon, but just like for our own mental well-being. You know, it's like we've lost so much of our forest. And the more we can do, just keep natural forest ecosystems just thriving, allow this natural selection. I just see it's just such a valuable tool. So I really hope that, we can really just put more effort into like building more forest, building more greenery, more, and just like having this better understanding of all the benefits that we get from it. I hope will inspire you all, because all this information really inspired me, and it just makes me feel really honored to be able to share it all with you. And that's just how I'd like to conclude right there. I just want to thank you all for sitting through this, and I really hope I inspired a few of you, and now you're thinking about things a little different way and seeing, the forest in a different way. You know, when you're walking out there looking at these mushrooms, like, are they degrading things? Are they associating with different tree species? And doing some research. It's so much fun just learning about these associations. So thank you all so much. I hope you really enjoyed that. This was really fun for me to teach. Enjoy the rest of the presentation on the Mushroom Summit. This is really exciting. Check the links below for my notes and my big Microforestry 25-page written paper. There's lots of awesome resources, all my cited citations. So, I'm not just making all this information up. It's coming from real scientific research. And I have these easy notes to follow along with and beautiful pictures for you to see. So, really encourage you to check out those resources.